So today we're just going to talk about gene expression. Okay, it's actually a very short topic. Okay, so this is gene expression and gene regulation. Okay, so these are the two main things that we're going to be talking about. So firstly, even before we go on explaining about what gene expression is and gene regulation is, what you need to know is that you need to understand what is transcription, what is translation. Okay, you need to have a background knowledge. That's the very reason why you learn transcription before we even learn gene expression. Okay, so our objectives today is that we're going to learn the importance of gene expression. So firstly, we'll just learn about the importance of gene expression. And two, we're going to learn to use the end product inhibition and the Lac Operon system. So we'll use end product inhibition, and in using this we'll use and Lac Operon as an example. Okay, so basically what I want us to do is firstly we need to understand what exactly is gene expression. Alright, so in our introduction today we're going to look at the regulation of gene expression. It refers to the, so basically it's a mechanism. Okay, it's a mechanism that cells use to switch the process of transcription and so it's a process that the cells use to switch the process of transcription and translation on and off. Okay, so just with this statement, this is where our center of attention is going to be. Okay, so the first thing we're looking at is that it's a mechanism. Okay, secondly, it's a mechanism that the cell uses to switch the process of transcription. Okay, sorry for that, transcription and translation on and off. These are our keywords. Okay, so having said this, we're going to look into details. How exactly does this happen? Okay, so gene regulation is important as it allows an organism to express certain proteins only when they're needed. Okay, so it allows, so this is the importance. It allows an organism to express certain proteins only when needed. Okay, so it means proteins in the body are not always transcribed. Okay, it's not all the time. So looking at this process, it allows an organism to express certain proteins only when they're needed in the body. For instance, the enzymes that are in your stomach. Okay, it's not all the time that they need to be there, or all the time that it's not all the time that they're there working. So they can only be expressed when there is need. Okay, and because of this, you find that the class it's like it preserves resources. Okay, so it avoids wastage of material and energy. Okay, so it avoids wastage of material and energy. Okay, so because the process of transcription right here and translation can be switched on and off, it means materials are preserved. Okay, so it avoids wastage of materials and energy, and it allows an organism to express certain proteins only when they're needed by the body. That's our main concern. Okay, all right. So having said that, so what you're going to see is that the classical example of a gene regulation system is of the lactose operon. Okay, so we're going to concentrate on the Lac Operon. That's the classical example of gene expression. Okay, so this Lac Operon is a classical example. Okay, it was discovered by Jacob and Monod. Okay, these are the two biologists that actually discovered the Lac Operon. So a Lac Operon is a system in which enzymes involved in lactose metabolism are expressed by the E. coli. Okay, E. coli is a bacteria. Okay, so they found it easier to actually discover what happens during gene expression by using bacteria. Okay, so I want us to note the key things that happen during the Lac Operon. Okay, so the E. coli was grown in the presence of either lactose or glucose. Okay, so either there was lactose or there was glucose absence. Okay, so they tested this in order to see what would actually happen to this bacteria. Okay, so before we go any further, are there any questions? Hello? All right, so what exactly is an operon? Okay, when we look at an operon, it's a group of genes which work together to control a single promoter. So I'll just write the description here. A group of genes that work together to control a single promoter. So what I want you to remember is, remember during transcription we have what we call the promoter site. Okay, so remember that this is related to transcription, and remember that gene expression is a system that cells use to turn on the process of transcription and translation on or off. So when you look at this operon, okay, it's a group of genes that work together to control the promoter. Okay, why are we targeting the promoter? Okay, why is it our center of attraction? It's because this promoter region is a site where transcription starts from. It's like a recognition site, like we learned during transcription. Okay, so for RNA polymerase to actually discover this is where transcription begins, it has to find where the promoter region is. That's why the operon is a group of genes that work together to control a single promoter. So the only way the gene can be switched on or switched off is by controlling the starting point. Okay, so these genes may be expressed or rather transcribed and translated together, and the protein products may have related functions. Okay, so don't worry about that, we're just going to look into it in more details. Okay, so the gene expression that's found in eukaryotes and prokaryotes, is quite different. When you look at the gene expression in eukaryotes, it's more complex than in prokaryotes, and its regulation takes place at different levels. Okay, so gene expression gene expression in eukaryotes, okay, is different in that it can take place during transcription of DNA into RNA. Okay, it can also take place on during cell differentiation. Okay, it can take place during growth and development. So this growth and development, this is actually a higher process, a component of embryology. As you go higher in your medical field, you actually learn about how a child forms every step. So this is how we have growth and development. Okay, it's actually more complex. So we also have translation of mRNA. Okay, so this is just to simply tell you that the process of gene expression in eukaryotes is more complex because it takes place at several stages in eukaryotic cells. Okay, so each of these steps represents a potential point. So these steps that I've outlined here, and few others that haven't been mentioned. Okay, so each of these steps that have been outlined, they represent a potential point through which the gene expression of eukaryotic genes may be turned on or off. Okay, so we'll first start talking about what we call upregulation. Okay, so upregulation is a process in which an internal or external results in an increased expression. So for instance, if this is a cell, okay, you have the nucleus there. So what they mean is that it's a process in which the internal, so whatever is happening inside the cell, or external signal results in increased expression. So whatever process is happening results in increased expression. Okay, that's what they mean by upregulation. All right, so it results into increased expression of genes, resulting into increased protein synthesis, because these processes occur, the process of transcription, okay, occurs in the nucleus, isn't it? So we have the DNA template strand, and we have the new RNA that is being synthesized, okay, which later on has to go out of the nucleus to be translated by the ribosome. Okay, so now what they mean is that the internal, whatever is happening here internally, okay, inside the nucleus, will result in an increased expression of genes. Okay, we are going to have more proteins being expressed. Okay, so this is to tell you that for a protein to be expressed, it's dependent on what is happening inside the nucleus. Okay, so they're just trying to tell you that you can't have translation without transcription. Okay, then however, when we look at down regulation, okay, down regulation, it's a process resulting in decreased gene and corresponding protein synthesis. So what you notice is that if in the nucleus the mRNA was not synthesized, it means there will be no translation. This will not occur, okay, because there was no transcription in the first place. So it's down regulation. Okay, all right, so the next thing that we're going to talk about is an inducible.
I have a question.
Yes, go ahead.
Oh, like it's not really a question, but I would like you to repeat on the up and the down, like the difference. I haven't really gotten it clearly.
Okay, so what I'm saying is that why you have up regulation. So what I said is upregulation is a process in which the internal or external signal results in increased expression of one or more genes, resulting in increased protein synthesis. So the best example I gave is uh, during protein synthesis. Can you help me connect? Okay, just a minute. Let me just connect the laptop. All right. So what I meant is during transcription, you notice that we have the DNA template strand, right? And we have the new messenger RNA which is being synthesized, and done being synthesized, it goes out of the nucleus in order to be translated. Okay, so it is translated by the ribosome. Okay, now for upregulation, they mean a process that results in what happens internally in the nucleus. Okay, and what is happening internally here, it's transcription. So if transcription happens and the mRNA goes out, then it means proteins are going to be synthesized. We're going to have what? Protein synthesis. But if transcription however does not occur, okay, it means there will be no protein synthesis, and this will be called down regulation. Okay, so down regulation is a process resulting in decreased gene and corresponding gene expression. So the decreased gene expression is here. Okay, it means the transcription did not take place, hence there was no protein synthesis. Does that make sense? But for
Yes,
upregulation,
it does. Thank you.
all right, thank you so much. So we're going to go into the next segment. So before I explain the inducible system, let me just highlight this. So when you look at an induced and repressible systems, okay, so I need you to be as interactive as you can be, because at some point it may get confusing if you're not really getting the concept. So how many who can explain to us what it means by when something is induced? Can anyone explain for us? Hello? Or if you've ever heard of something like induced labor. All right. So what we mean by saying an inducible, actually it's supposed to be inducible. Okay, so this is inducible or repressible system. So an inducible system is normally switched off. So key things you note here is one, it is normally switched off. Okay, except in the presence of an inducer molecule that switches the system on. Okay, so what you note here is that presence of an inducer can switch on the system, can switch on the system. Okay, so what I want to explain here is that if something has the capacity to be induced, okay, this word induced, it means you can it has the ability to be switched on. Okay, now for instance, let me give an example of, in a house, right, you have a switch at the side of the wall where you have a bulb, right? This bulb is normally switched off, right? So this bulb is on a normal regular base, it's normally switched off. Okay, so follow me, follow through with me. So it's normally switched off. Okay, but what you notice is that sorry, let me just erase this. Okay, so on a normal regular base you find that it is normally switched off, but the presence of an inducer, so you when you come in the house, okay, or you go in your room or in your hostel, you're the inducer, so your presence there is what is going to switch on the system, and the system is this bulb right here. Okay, so an inducible system is normally switched off. So you find our system here is the bulb. So on a normal regular base, it's normally switched off, except in the presence of an inducer, which is you. When you go in the room, you switch it on. Okay, you turn it on and the bulb goes on. Okay, so the reason why they call it inducible, because it has the ability to be switched on, though it's usually switched off, but it can be switched on. Does that make sense? Hello? Hello? Hello, are you following? Okay, have you understood this point? Hello? Alright. So, now that we've talked about an inducible, we're now going to talk about a repressible system. Okay? Alright, so what you notice here, so a repressible system is normally switched on. It's normally switched on. Okay, but can be switched off it can be switched off, okay, in the presence of a co-repressor molecule. Okay, so in the previous example, co-repressor molecule that suppresses gene expression. So the example I gave you of a house, okay, so you have this house. Now in this case, I'm going to consider the bulb as the bulb is on, right? It's emitting light. So if this bulb is always on, does it mean it can't be switched off? Hello? 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 Okay, so I'm asking a question. If you go in a room and you find the bulb is off, I mean it's on the whole time, does it mean it can't be switched off? It can be switched off, right? Okay,
Yes.
thank you so much. So that's what they mean. They mean it's repressible. You can switch it off. On a regular base, it's normally switched on. okay, but it can be switched off by the presence of a co-repressor. So the co-repressor is you when you come in the place, okay, when you come in the house or room. When you switch it off, then the system can be switched off. So what exactly is trying to be explained here is that gene expression can be induced or repressed. It can be switched on or off. Okay? Now, if it's inducible, it means it's normally switched off, but it can be switched on in the presence of an inducer. Okay? But when it's repressible, when it's a repressible system, it means it's normally switched on, but it can be switched off in the presence of a co-repressor molecule. Okay, does that make sense? Are you able to connect the reason why we say the Lac Operon is a group of genes that work together to switch on or off gene expression? Or rather to control the single promoter. Are there any questions on what we have discussed so far? Okay, if there aren't any questions, we'll just go straight to talk about the Lac Operon. Okay, so when you look at the Lac Operon, it has 3 structural genes. It has 3 structural genes, okay, which can be controlled by a regulator gene, okay. It also works in conjunction with the promoter sequence. Like we said, the Lac Operon works because it's a group of genes that is there to switch on or off the promoter. It works with the promoter region, is it? Like we discussed previously, they work together to control a single promoter. So we have it works in conjunction with the promoter sequence. Okay. The other thing, it works in conjunction with an operator sequence and terminator sequence. Okay, so promoter sequence and terminator sequence. Okay, so one thing about the Lac Operon, it has 3 structural genes which we're going to look at. It has a regulator gene, it has a promoter sequence, it has an operator and terminator sequence. Okay, so now we're going to go in details to look at this. So the 3 structural genes that are in the Lac Operon, we have LAC-Z. We also so this LAC-Z, and please, it's very important that you know what this LAC-Z encodes. So it encodes the enzyme. So LAC-Z encodes the enzyme beta galactosidase. So beta galactosidase or galactosidase. Okay, so what's the function of beta galactoside? What it does is that it breaks the disaccharide. Okay, it breaks the disaccharide lactose into, so what 2 molecules make up lactose? Anyone to try? Hello? Hello? So it breaks the disaccharide lactose into glucose and galactose. Okay. Another thing that we have is we have Lac Y. We have the Lac Y gene. Oh, sorry. So we have our Lac Y gene. Okay. This Lac Y gene encodes for the membrane-bound protein beta-galactoside permease. So it encodes for a protein called beta-galactosidase permease. Okay. Okay. Alright, so when you look at this beta-galactoside permease, so to permeate is the ability to pass through something. Okay, that's why we're always talking about, oh, the cell membrane is permeable because it allows substance to pass through it. So what Lac Y gene does is that it enables lactose, or when it's broken down into glucose and galactose, in order for this glucose to be absorbed by the cell, it needs this beta-galactosidase permease. Okay. Okay, so it pumps lactose. Okay, it pumps this lactose into the cell. That's where the permeability is coming from. Then the other thing that we have is, we call it Lac A. Okay, Lac A gene. What does Lac A do? So Lac A encodes for the enzyme beta-galactoside transacetylase. Okay, transacetylase. So this is the enzyme. Okay, so this enzyme modifies the lactose. So it modifies lactose. Okay, so it's very important that we talk about these structural genes in depth so that you're able to understand what exactly is happening. So the Lac Operon is required for the efficient transport and metabolism of lactose into the E. coli. Okay, in order for the gene to be expressed or not, we need these structural genes. Okay, and some related bacteria. So in the presence of glucose, and before I go to that, just want to have a recap. So we talked about Lac Z, okay, which encodes for the beta-galactosidase, and this beta-galactosidase breaks the disaccharide lactose into glucose and galactose. Then we have Lac Y, which encodes for beta-galactosidase permease, which pumps lactose into the cell. Okay, and the next thing you talked about is Lac A gene, which codes for beta-transacetylase, which modifies the lactose. Okay, so the next thing that we talked about is the Lac Operon in more details. Okay, so in the absence of glucose, okay, the cell can use lactose as an energy source. So I want you to imagine this on a normal regular basis. When you have glucose in your body, okay, glucose can be used for the process of respiration, okay, in order to generate ATP, right, which is energy. Now what they're saying is that if glucose is not there, lactose can be used instead, because this lactose, when it's broken down, we have galactose and glucose itself. Okay, so the lactose is like a backup. All right, So in the absence of glucose, where you don't have glucose, the lactose can be used to provide glucose, which can be used in the process of respiration. Okay, so here we're going to be working with lactose and glucose just to get the whole concept of gene expression. And remember, this gene expression was being done on bacteria. So what you notice that in the absence of glucose, the cell can use lactose as a source of energy, like I mentioned, but it must first produce the enzymes that are needed to mobilize. Okay, those enzymes that we're talking about, the Lac Z, Lac Y, and Lac A. Those enzymes are needed to modify the lactose, okay, and to mobilize it in order for the body to digest the lactose. Okay, so the lactose cannot just be absorbed by the body directly as it is. It has to be modified, or rather mobilized. Okay, are we clear up to this point? Hello? Alright, so the Lac Operon ensures that the cell uses energy in producing the three enzymes, okay, when necessary. So how does the Lac Operon ensure that it has energy to produce these enzymes that are encoded by this Lac Z, Lac Y, and Lac A? It achieves this by using the repressor molecule, okay, so it uses the repressor molecule. So we'll talk about it more in details. The repressor molecule stops the production of these three enzymes in the absence of lactose. Okay, so the repressor molecule has two active sites. So in order for you to understand this, let me just have something that's diagrammatic that will help you understand. So the Lac Operon is something like this. I'm sure if you've seen it in your lecture notes. So we have Lac I, and by the way, Lac I it encodes for the repressor molecule. We also have the promoter region, which is labeled P. We have the operator region, like I mentioned in the first place. Then we have the three structural genes, Lac Z, Lac Y, Lac A. Okay, they follow each other sequentially like that, and the termination. All right, so what that's saying is that this is our DNA, right? This is our DNA, and remember, this is happening in the what? In the nucleus. Okay, so if this is your gene and you want to transcription to occur, and we want to either switch it on or switch it off, we're going to see how that happens. So what happens is that in the absence, the repressor molecule, sorry, before I talk about that, so how it gets energy to transcribe these, you find that in the presence of in the absence of lactose, okay, we have the repressor molecule. So this repressor molecule is encoded by a gene, okay, which is called the Lac Y. So when this repressor molecule, okay, so this is the repressor, and remember, for all these things to work, they need first to be transcribed and translated. So this is mRNA, okay, and then when translation occurs, we have our repressor molecule. Okay, so what happens is that how does this repressor molecule stop transcription from happening? So remember, right now we don't have lactose, right? This is absence of lactose. So what happens is that the repressor molecule has two sites. So let me draw it like this. Okay, it has two sites. There's this site and the other site. So so one site can either be the inducer site or the other site can be the repressor site. Okay, so the repressor has two sites in which it can either be an inducer molecule for lactose, or it may be attached to the operator gene, or a co-repressor glucose molecule may be attached to switch the operator gene off. So it has two sites, one for the inducer, which is the lactose, okay, which can switch on the system. So you're going to see how the system can be switched on, or for the glucose. Okay, now how exactly does this happen? So because it has two sites, okay, these allosteric sites, okay, I'm sure as you learned about proteins, you know that proteins are, they are specific, they're like enzymes sort of, okay, so they have a specific site. So what happens is that in the absence of this lactose, where lactose is not present, it means glucose is present. Okay, and this is logical enough. If you have enough glucose, there's no need for lactose. Okay, so when you have enough glucose in the body, what it means is that you not need these three enzymes to be transcribed, because these three enzymes are going to go and mobilize the lactose in order to give you glucose again. But you know that in your environment you already have glucose. So what does glucose do? Glucose goes and attach itself, okay, so it activates the repressor molecule, and when the repressor molecule gets activated, this repressor molecule goes to bind on the promoter site. Okay, now when this, let me just label it as R, when the repressor molecule binds on the promoter site, it prevents the binding of RNA polymerase. Okay, so what happens is that when the repressor molecule binds on the promoter site, it disfigures the shape of the promoter site so bad that RNA polymerase can't even bind on this promoter site. Okay, and because of that, if RNA cannot bind on the promoter site, it means these three genes will not be expressed. Okay, and transcription has been switched off. Okay, does this make sense? Hello? Okay, thank you so much. So the opposite is true. So let me just explain what happens. What can happen when now we have, when we have no glucose. Okay, so take for instance, you've been fasting or you haven't eaten something the whole day. Your body has less glucose. What will happen? Lactose is needed, right? Because lactose can be used to provide the body with energy. All right, so when lactose is present, it means it's an inducer. It can turn on the system. Okay, so what happens is that lactose goes to bind on the inducer site on the repressor molecule. Okay, so because this repressor molecule has two sites, active sites, it means when lactose binds here, it distorts the shape. Okay, so it would distort the shape of this repressor molecule. Okay, so if the shape of the repressor has been distorted, it means even when it comes to bind here, if the shape of the repressor was like this, you find it to be something else, such that it will be unable to bind on the promoter site. Okay, and because it's unable to bind on the promoter site, it means the promoter region is now going to be free of the repressor. Hence, RNA polymerase can come and bind and transcription can take place. And all these three genes, LAC-Z, LAC-Y, and LAC-A, are going to be transcribed in order to make what? In order to modify the lactose, and by the end of the day what we want is glucose, which was initially not present in the beginning. Okay, so in this process where you have lactose binding to the repressor molecule, okay, the whole point is that when lactose comes to bind here, it will distort the shape, and when the shape is disfigured, it means the repressor can no longer bind on the promoter site. Hence the promoter site is going to be left open, and RNA polymerase can now come and transcribe the transcription. So it means that this system is turned on. Okay, do you understand? Hello?
Yes, madam.
Okay, so the repressor molecule, like I said, because it has two active sites, it may be attached to switch the operator gene, or co-repress, which is the glucose molecule, may be attached to switch on the operator gene off. So when the operator gene is switched on, the structural genes can carry out transcription. So sorry, let me just correct this. So when you look at this, we said we have the promoter region and the operator. So what it is that what is supposed to be targeted is actually the operator region. Okay, so sorry for the mistake. So this operator region is what the repressor targets. Okay, so if the repressor molecule attaches itself here, it means when RNA polymerase comes here, it will be blocked in a way, because this whole place is going to be disfigured. It can no longer transcribe this. And the same is true when this operator site has not been tampered with. It means RNA can now transcribe. So I just wanted to correct this mistake of saying it's on the promoter. It's actually on the operator gene that all this happens. Okay, okay, so all this is happening on the promoter, on the operator. Sorry, this is the operator. The operator site is the one that is being targeted. Okay, it can be switched on or off depending on what binds on the active sites. So are there any questions? Hello? 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 Hello?
No, madam.
All right. So having
No question.
okay, thank you so much. So what I just want to clarify with you guys is that the whole process of the gene expression, it's to either switch on or off translation or transcription. Okay, so like you can see, this is the Lac Operon. It's a group of genes that are working together. Okay, and that are getting the main target is the promoter site, because they know that if we block this part, the RNA polymerase will not attach itself on the promoter site, which is the starting point of transcription. Okay, so the Lac Operon, when it's switched on, it means these three genes are going to be transcribed: LAC-Z, LAC-Y, and LAC-A. Alright, these are going to be transcribed in order to work on the lactose. But if the system is switched off, the three genes will not be transcribed. So this takes us back to the function of the Lac Operon, where we say it's used to conserve, or rather preserve, resources. Okay, and also it enables the system to be switched on only when certain proteins are needed. Okay, is that okay? Hello?
Yes.
Okay, So we're going to talk about the last things. So we'll talk about the Lac Operon in the induced state and in the repressed state. So the Lac Operon in induced state. Okay. So when the lactose, which is the inducer, so remember, lactose is the inducer. Okay. So when the lactose, the inducer is present, it alters the shape of the repressor. Okay, like I said, it's going to alter the shape of this repressor when it binds there. Okay, which then can no longer bind to the operator. So there'll be no binding to operator, to the operator region. Okay, so it means the genes lac Z, lac Y, and lac A will not be transcribed. Okay, so this is when the Lac Operon is in the induced state. Okay, and like we said, an induced state is normally what? Switched off. Okay, but it can be switched on in the presence of an inducer. So now here we're saying that this induced state is it's switched off. Okay, sorry for that mistake. So it's this induced state is switched off. Okay, why is it switched off? It's because lactose, which is the inducer, when it's present, okay, when it's absent, sorry, when lactose is absent, it means when it's present, yeah, when lactose is present, it can bind on the repressor molecule, and hence gene expression will not occur because the repressor molecule will not bind on the operator region. Okay, so you can marry these to the induced state with the inducer. So when it is in the induced state, okay, when something is inducible, it means it can be switched off. Okay, so this induced state has the ability to be switched off, or rather it has the ability to be switched on. So it can be switched on. Let me just clarify on this. So if something is inducible, like we said in the first place, okay, let me just take you back here. Okay, so we said the inducible system is normally switched off. Okay, so because it's normally switched off, it means it has the ability to be switched on. So this inducible system is normally switched off, but it can be switched on. Okay, so it's inducible because it's normally switched off. Okay, and why is it switched off? It's because when there's the presence of lactose, which is the inducer, it goes to bind on the repressor site, on the repressor molecule, which has two active sites, one for the repressor and one for the glucose. So now when it binds here for the inducer, on the inducer site, it distorts the shape, and hence the repressor can no longer bind to the operator region. Hence the genes like Z, Y, and A will not be transcribed. And the other state of the cell is when the cell is actually in its repressed state. Okay, we're also going to look at when the cell is in its repressed state. So repressed state. So when it's in its repressed state, the repressor protein binds to the operator site. Okay, so when it's in its repressed state, it means what? The repressor, so they just go hand in hand. So in the repressed state, so let me go back to this. before I go to the repressed state. Okay. Alright, so in the inducible state, we said the system is normally switched off, but it can be switched on when there's an inducer. So like you saw when the lactose was present, it means when lactose is present, the system can be switched on. Okay? Because normally it's switched off, but now it can be switched on because there's a presence of lactose. Now in this case, in the repressed state, where you have the repressor, okay, it means glucose is present. Okay, so when glucose is present, it means glucose goes to bind on the repressor site and it gets activated. Okay, when it gets activated, it means this repressor site, this repressor molecule, sorry, the repressor molecule, sorry, don't mind the drawing. So what it means is that this repressor molecule can bind to the operator region. Okay, so like Z, like Y, and like A, and this is the terminator. So we have the promoter here and the inhibitor. So when the repressor binds here, it stops the RNA polymerase from even going forward because it disfigures the shape here. So hence these three genes will not be transcribed because the presence of the repressor. Okay, but in the inducible state, what you notice is that when we have lactose, the inducer, it means the repressor can no longer bind on the operator. Hence, these like Z, like A, and like Y will be expressed. Okay, because the inducer is available. Are we clear on this one? Are there any questions? Hello?
No.
Oh, all right. Okay, so the end product inhibition is our last thing that we're going to talk about. So we have what we call end product inhibition. So this end product inhibition occurs when the E. coli is grown in a, or rather on a glucose medium. Okay, so we have the bacteria here. Okay, the bacteria has no nucleus. Okay, so this is the bacteria, and what you notice is that it's being grown in the glucose medium. So this is the glucose medium. Okay, so just picture this is put somewhere where there is glucose. So the regulator gene produces the repressor molecule. So what you notice is that we have this Lac Operon that we have talked about. We have like I, P, promoter, operator, then our structure genes Y, Z, and A, and the terminator site. So where there is glucose, what happens is that the regulator gene produces the repressor molecule, okay, which binds the glucose. Glucose is a co-repressor. So when they say glucose is a co-repressor, they mean it helps to switch off the Lac Operon. Okay, so if you're colleagues, it means there's a connection between two things. So glucose is a co-repressor because when it binds to the repressor molecule, it helps it to stop, or rather to switch off the Lac Operon. So the Lac Operon gets switched off. Okay, So in this is a case of the end product inhibition or repression. It is referred to as a negative feedback. So it can be referred to as a negative feedback. Okay. Why is it referred to that? It's because our end product, which is the glucose that we actually want for energy, is being used to prevent the process, or rather to switch off this process. So what we're trying to say is that if your body has enough glucose, okay, enough glucose, this end product, okay, this glucose, when it's too much, or rather when it's enough in the body, there will be no need to create more glucose. Hence, the very glucose that you have in your body is going to be used to prevent more glucose from happening, okay, by binding to the repressor molecule. That's why it's called negative feedback. Are we clear?
Yes.
Yes. All right. So we've come to the end of this topic. The next time we'll be talking about mutagenes. Then we'll go straight on to talk about mutations. So are there any questions?
Maybe we should try to send some questions based on the same topic.
Okay, so I was actually planning that we should do a tutorial together. But if you insist, I can make it as an assignment to see how well you have understood. So just go through the notes, and with what you've learned, I'm sure you'll be able to cement. Okay, so are there any questions? All right, if